This video will discuss in depth and provide a conclusive understanding of recent literature related to iron metabolism in dementia. Dementia is the single greatest cause of disability in Australians aged above 65. There are currently 459 Australians living with dementia, and almost 50 million people worldwide are affected by the disease. Every 3 seconds, someone in the world develops dementia, a figure that is expected to rise by more than 25% within the next 5 years. Dementia is also the second leading cause of death to Australians, with females accounting for 64.5% of all dementia-related deaths. Dementia is an umbrella term for a number of specific medical conditions that trigger progressive decline in cognitive abilities. These changes include loss of memory, intellect, social skills, rationality, and physical functioning. The main causes and types of dementia, from the most common to the least common, are Alzheimer's disease, vascular dementia, Lewy body dementia, frontotemporal dementia, Parkinson's, Huntington's, alcohol-related, and mixed dementia. Iron is the most abundant trace element in the human body and is an integral component of hemoglobin involved in the transport of oxygen within the body. Some main sources of iron include dried beans, nuts, spinach, red meat, and chicken. The healthy population require approximately 1 to 2 milligrams of iron per day to maintain iron homeostasis within the body. The body metabolizes iron through the duodenal and upper jejunum enterocytes into the intestinal epithelial cells, where it can be stored as ferritin or transported into the hemoglobin inside red blood cells to be circulated around the body. As blood circulates around your body, iron is transferred into the brain across a blood-brain barrier. This barrier prevents toxic compounds from entering the central nervous system and removes waste products from the brain tissue, while still allowing essential molecules such as iron to selectively enter the brain. Transferrin receptor 1, expressed on the endothelial cells of the blood-brain barrier, mediates iron uptake in the brain. Transferrin is a blood plasma glycoprotein which plays a fundamental role in iron metabolism and is responsible for ferric iron, oxidated iron delivery. Dementia patients, in particular those with Alzheimer's, have lower levels of iron in their plasma when compared with healthy subjects, and according to a study conducted by the Alzheimer's Association in 2010, the high anemia levels found in subjects with Alzheimer's disease was not explained by dietary iron deficiency. The Australian Mental Health Research Institute in 2010 also examined the Australian study of ageing, concluding that disturbed brain iron metabolism was reflected in the rest of the body by a decrease in plasma iron and hemoglobin levels. This systematic deficit in iron transfer within the body could contribute to morbidity associated with dementia, as the catalytic properties of iron are essential for a number of metabolic processes. In 2015, Hare et al. Conducted a study showing that while transferrin levels in both the healthy and Alzheimer's subjects were similar, iron-bound transferrin was significantly decreased in Alzheimer's subjects. Therefore, they hypothesized that the transferrin glycoprotein in people with Alzheimer's disease was not working properly to metabolize iron, thus causing iron dyshomeostasis within their brains. This then creates a buildup of iron in the brain, which is a feature of normal aging. However, when coupled with a loss of iron homeostasis, neurotoxicity can then occur, which causes neurodegenerative diseases such as dementia and Alzheimer's disease. In 2019, Chang published research giving light to the fact that iron accumulation may be related to the metal binding sites of beta amyloid, causing formation of plaques between neurons in the brain and abnormal tau protein accumulations. These deposits may be related to abnormal expression of various brain iron metabolism-related proteins in the brain of Alzheimer's patients, and the difference is abundant in this image comparing microscopic images of a healthy brain compared with a brain affected by dementia. Furthermore, this MRI image shows an image of an Alzheimer's brain compared with a healthy brain, showing a buildup of white matter, which has been suggested to be indicative of myelin loss. In the brain, iron is the key substance involved in the myelin protein synthesis. Therefore, when iron is not in a state of homeostasis, this has a trickle-on effect, causing multiple neurodegenerative issues. Furthermore, Alzheimer's disease is a multifactorial neurodegenerative condition associated with pathological accumulation of amyloid plaques and tau. When these proteins accumulate within the brain, it disrupts communication between brain cells and active immune cells. The immune cells then trigger inflammation, which then ultimately destroys the brain cells. Conversely, in 2014, Crespo et al.'s study indicated that the key proteins controlling the transportation and storage of iron in the body, transferrin and ferritin, were significantly decreased in the serum of Alzheimer's subjects than in the control group. Therefore, while the available data about both brain and circulating iron levels have identified a measurable change, the data is very heterogeneous and inconsistent, requiring further research and clarification. Additionally, as the brain iron accumulation manifests at regional, cellular, and subcellular levels, it would be beneficial to understand both the cellular and intercellular transport mechanisms within the brain. On top of this, understanding the exportation of iron from the neurons to other parts of the brain needs further clarification in order to determine reasons for the iron accumulation as well as prevention methods. As discussed, research is currently inconclusive surrounding iron metabolism in dementia. However, although it is inconclusive, there are still various potential explanations of the role iron metabolism plays in dementia development, meaning research is on the right track, however still needs further funding and in-depth studies.